of course, we go to the mechanism. So the first one we already talked about is unequal crossing over, and we explained earlier in this lecture how unequal crossing over can increase the DNA contents in the genome, and the unequal crossing over can happen in any places in the genome or in the gene region. So in this figure, actually it shows the unequal crossing over happen within a gene. So in this case, you can see the new genes in the middle. Now it's a chimera, has half the gene content from the green and half the gene content from the white one. So it's a chimera genes in this case. But the unequal crossing over can happen in any places. And the B case is somewhat ignored for a long time because we didn't recognize it's an important mechanism until 1990s. So this mechanism is by retrotransposition. So as shown in B, you know, in our genome, when the gene, especially a protein coding gene, we first express the messenger RNA. So we have the mature messenger RNA produced in the genome, and this matured messenger RNA contained only the information from the exons, all the introns were spliced out, and also have specific processing, have a cap in the 5' end and add the long poly A tails. So, and this transcript will be sent to the cytosol for translation. So this is your molecular biology. But if in the genome we have retroviruses, so a lot of those RNA viruses, they have an enzyme who can convert RNA molecule to DNA. So by chance, they use our messenger RNA as template. They will reverse transcribe our messenger RNA into a complementary DNA sequences. So that's the cDNA. Then if this cDNA insert into our genome and acquire the local sequence for its transcription, and this can be a new gene in our genome. Why this was ignored in the early days? Because first, we didn't know there's an enzyme called reverse transcriptase. You know, this discovery was revolutionized our central dogma. Second, we think the transcriptional regulation, although in early days we know very little, but by understanding how lactoperon is controlled in bacteria, we know transcriptional regulation is quite complex, even in the prokaryotic genome. So in eukaryotes, it's very, very unlikely a DNA sequence randomly inserted into the genome and can be expressed right there. So because you know it's not only the coding sequence itself, showing here is the cDNA region. You need a promoter, you need some transcription factor binding site to activate the transcription. So traditionally, we think retrotransposition is not going to produce any new gene material. They will randomly insert a gene and stay there and degrade. So that's all we think about retrotransposition. But this view was totally changed by Professor Wang Yuanlong's research. Before we talk about retrotransposition in the genome or other type of duplication mechanisms, I want to talk about a little bit fate of gene duplication. Fate of gene duplication, classical view is if a DNA or a gene gets a duplicate. I already introduced you some of the early wisdom. They say, oh, so they will be relaxed from selection because mutation happen at this region. You always have a good copy. So the bad copy is not going to hurt you much. So that's our view. And this view leads to most our thinking are the duplicate copy most likely will become a pseudogene. That means the gene has no function because the mutation accumulation. So that's one of the most sort of like a belief. But as I told you, from the DNA content and gene number, so we count the gene number, we know Drosophila has more genes than yeast, and human has more genes than Drosophila. So we do have more genes for more complex multicellular organisms. So in that case, how do we explain why we have so many active duplicate copies? So this was proposed by Force in 1999 in genetics, and he is research staff in Michael Lynch's group. So they proposed there are 3 possible ways to think about a duplicate copy, a fate of duplicate gene. The first one is what I just mentioned, the pseudogenization, means the gene goes to a non-functional copy. So one of the copies. So now after gene duplication, you have 2 copies of the same gene. So any one of them can go non-functionalization without affect the individual's fitness. The second one is the neo-functionalization, and the third one is the sub-functionalization. So for the other 2 fates, focus on the change at the regulatory sequence. Okay, so showing this slide, the long black bar is the coding sequence, but the small square or triangles or other triangle and squares are the regulatory sequence of the same gene. So this is like how they want to illustrate their idea. So for neo-functionalization, it means the duplicate copy, so then you have 2 copies. Any of these 2 copies might acquire new function by change its regulatory sequence. When I say by changing its regulatory sequence, actually what I meant is the mutation occur at their cis regulatory region might lead a new function, for example, an expression at a different tissue or a different developmental stage. So that was showing by the triangle in this middle part of the slide. And the third one is very interesting. Ideally, if a gene expressed in multiple tissues or multiple time during development, then we have several different enhancer, or we say cis regulatory modules. Showing here, there are 4 cis regulatory modules showing on the top. So this gene has 4 cis regulatory modules. They are all important for its expression. After duplication, is like a whole gene duplication. So both copy contain the full-length coding sequences, also the entire cis regulatory elements. So this is a very ideal situation. Then now, after the duplication, mutation could occur randomly. If a mutation occurs at the cis regulatory region of one of the copy, now showing in white. In this case, if this is important function for this gene for the animal's fitness, then this mutation preserved the other copy, especially for this particular cis regulatory region of the second copy. Now, any mutation happen at that region will probably very deleterious. The individual won't live in the population. So that's why all the live individual has a functional copy of this cis-regular element on the second copy. So that's why a mutation happens in one of the copy preserve the function of the other copy because the other copy cannot go bad. If this is the case, you can think about the two copies could diverge in their function by the third process called subfunctionalization. Think about the GPI in fish. Now the enzyme expressed at several different tissues, and also they differentially express at, for example, I just randomly pick up a liver and muscle. So now one of the copy expressed in the liver, the other copy now expressed in brain, muscle, and other places. Both copies are important and both copies are essential after this subfunctionalization. And this is the idea combined everything we know about gene regulation, duplication, and evolution. So this was proposed, and this paper was like immediately recognized by people in several different areas. So the three fates, they state clearly, could be equally important for gene duplication. And we cannot just simply say a duplicate copy now have a chance or more chance to be degenerated, because there are other possibilities, and they try to understand how this could affect the genome complexity and the number of genes in our genome. Okay, so this is another illustration about the subfunctionalization or differential expression by Zhang's review paper. So this is also using different colors to show the two cis-regulatory modules now in the oval shape, so A1 and A2. Originally, the ancestral copy expressed in two different tissues, tissue 1 and tissue 2. Now, but after gene duplication, they can be specialized because the mutation occur. So this is like if mutation occur in one of the cis-regulator module shown here in green, then it becomes a white, a non-functional one. So now after this mutation happened, this copy now only expressed in tissue 1. And also now if this gene is an essential gene, now the expression in tissue 2 is completely relies on the other copy. So this is like explain how the differential expressed copies may maintain in the genome. And this is widely seen in variety of gene families or duplicate genes.